With our electrical potential, we have plus one, we have Faraday's constant, and then we have our voltage or our delta psi. This is going to be millivolts converted to volts to be reconciled or to reconcile with our units. And that's going to give us an electrical potential of negative 5.79. So these are both going to be negative values. So they're both going to be favorable. So this is favorable from a chemical perspective as well as a charge perspective. Now, that's going to give us overall negative 12.21. If we're to flip it and do anything different, well, this was we're transporting sodium into the cell. What about when we transport it out of the cell? Okay, what changes? Well, when we're transporting it out, our destination is going to change. So rather than, yeah, that destination is going to change, so too is the source. So rather than it being 12 over 145, it's going to be 145 over 12. When you do that, you're going to have a chemical potential difference that is unfavorable. An electrical potential difference, lo and behold, is actually also going to be unfavorable because of the fact that everything that you were doing, now we have to change our delta psi value. It's not going to be negative 0.06, instead it's going to be positive 0.06. So we're making this less favorable for a couple of reasons. Now, if all of this, like if you switch this from transporting sodium, let's say, rather than sodium, we were doing Cl minus chloride. Well, then this chemical potential calculation, that wouldn't change. That would still be unfavorable. The electrical potential, however, would be favorable because this would be a negative value. So that's going to make this a negative value. So when you see this sort of calculation or this sort of question, I strongly recommend that you break it down into two different parts, the chemical potential and the electrical potential. If you do that, you're going to be in good shape for these sorts of calculations. Okay, so there we go. Now, when we're transporting substances across or through a membrane, the transport may either be mediated or non-mediated. So we can break this down into passive transport and active transport. Passive transport can fall into the category of being mediated or non-mediated. Non-mediated would be this right here, diffusion through a lipid bilayer. Simple diffusion through a lipid bilayer. Now, the rate of that diffusion depends on the concentration gradient and the substance's solubility in the membrane's nonpolar core, which is an important thing to remember. Within that lipid bilayer, that's a nonpolar region. So you might have something that, yeah, can make it here or can make it across, but then once it gets into this space, it might really not like it, or it might be super comfortable there, in which case it's going to stay. Now, water, oxygen, and steroids can all diffuse across a membrane readily. Basically, anything else is going to have a very difficult time doing so. Now, in metabolism, you'll learn about some substances that can move across the membrane easily, but but there are not many, and yeah, it's kind of rare. Mediated transport. There are two categories depending on the thermodynamics of the system. There's passive mediated transport, also known as facilitated diffusion. Molecules are going to flow from high concentrations to low concentrations. There's also active transport. A molecule is transported against its concentration gradient. This endergonic process must be coupled with an exergonic process to make it thermodynamically favorable. So to move these green squares, A to position B, well, you have to couple that with something like ATP hydrolysis. Now, if we look at this passive transport. We've got diffusion through a lipid bilayer and facilitated diffusion. The difference between these is that the composition of those compounds are going to be different. So these nice little, I mean, they're orange triangles, so they just make me think of nacho cheese Doritos. Those Doritos are molecules that they're really not going to like going through that membrane. So they're going to have a protein that assists in that. Simple and facilitated diffusion. For both simple and facilitated diffusion, solutes only move in the thermodynamically favorable or favored direction. But proteins may facilitate transport, increasing the rate of transport. So if we think about simple and facilitated diffusion, and when we look at facilitated diffusion, what this looks like and what you should see is that we've got velocity versus substrate concentration, which is a Michaelis-Menten plot. So these proteins are going to, so this looks like a Michaelis-Menten plot, and whenever you look at the facilitated diffusion line, if you think about facilitated diffusion, going back to our previous slide, facilitated diffusion is protein dependent. There's an enzyme, there's a protein that's helping something move across. And what you notice about that is a lot like our Michaelis-Menten plots for enzyme catalyzed reactions. At some point, you increase your substrate concentration, and what happens to your velocity? Well, it's going to slow down. Or not slow down, I'm sorry, but it's going to effectively plateau because at some point, your gatekeeper is the concentration of your enzyme. Whereas with passive diffusion, as the substrate concentration goes up, the velocity goes up. And that continues to be the case. And that goes on into oblivion. Um, Now, important distinguishing features of these: facilitator requires assistance generally from a protein, and transport displays saturation kinetics, as I was just saying. Now, once saturated, the only way to increase transport with facilitated diffusion is to increase the number of transport proteins. Now, with transport proteins or with mediated transport, proteins are going to facilitate the transport of the molecule. This increases the rate of transport, and solutes can either go along their concentration gradient through what we know to be facilitated diffusion, or they can go against their concentration gradient in what is known as active transport. Now we've got three major types of transporters. We have a uniport, which is going to move one molecule across a membrane. We have a symport, which will move two molecules in the same direction, hence the arrows pointing in the same direction. Then we've got an antiporter, which moves two molecules, one and two, but in opposite directions, hence the arrows pointing in opposite directions. These are going to exhibit Michaelis-Menten saturation kinetics, and so you can evaluate a transport protein using a Lineweaver-Burke plot, as we show here. Now, there are also proteins that are known as channels or carrier transporters. Channel transporters have a pore through which the solute passes at a very high rate. Now, these show high selectivity, rapid flux rates, and tend to be gated. So when you see that word gated, I always think about a gate like I would think at a ranch, something that closes, something that basically is going to keep some things out. So now, some examples for these are porins, gap junctions, and ion channels. Carrier transporters bind the solutes on one side and undergo a structural change, releasing the solute on the other side. So we see that with one. So this this carrier proteins are right here. Think of them as basically having three different kind of phases or permutations. This is number one, this is number two, and this is number three. So essentially, if we look at phase one, that's where the molecule is open or the protein is open to receive basically a binding partner substrate from the extracellular space. It binds it, and then essentially what it does is it kind of closes off and reopens to deliver or deposit that molecule to the other side of the membrane. Um, GLUT transporters, molecules that are proteins that are responsible for transporting glucose into a cell from the bloodstream, for example, they function in this way where they basically receive glucose, close off, and then release it into the cell. These are divided into pumps and transporters. These are also known as translocases or permeases. Now, carrier transporters show temperature dependence, while channel-forming transporters do not. Now, there are drugs that are transport inhibitors. Transporters can make excellent drug targets. One example are serotonin reuptake inhibitors. These are common, or these are sometimes referred to as SSRIs, so selective serotonin reuptake inhibitors. Um, and what they do is they basically prevent serotonin from being taken back up by a neuron. Um, these transport inhibitors can cause death. Uh, Carboxy atricycloside and bongkratic acid inhibitors of mitochondrial ADP ATP translocation is how they function or what they do. Now, a selective serotonin reuptake inhibitor. So here's what we have is a neuron. Now, what this neuron is going to do is it's going to metabolize tryptophan into serotonin. Now, serotonin will be packaged into vesicles, and these vesicles that hold a bunch of serotonin will merge with the membrane of a neuron, and they will go through a process known as exocytosis. So this vesicle, which binds with the plasma membrane, will open up. These serotonin molecules will be released into the synapse, the region between or the space between our neurons, and they will bind to serotonin receptors. Now, these serotonin receptors will be stimulated and cause this receiving neuron to initiate some sort of change. Then eventually, these serotonin receptors will release that serotonin, and then a transporter protein on the originating neuron will enable those or will allow those serotonin molecules to go back into that original neuron. Now, in SSRI, what it's going to do is basically block this. So it's going to block that serotonin from going back in, which means effectively that this serotonin that's released, well, it's going to stay within the synapse. But in addition to that, this neuron, this original neuron, will just be like, oh, I haven't gotten a signal that I don't need any more serotonin, so I'm going to keep pumping serotonin into here to go onto these adjacent neurons. Now, porins are a pretty cool class of proteins. These are transport proteins, and this is an example of a porin. And it's this region right here that is the pore that's being made to allow for a molecule to go through. Now, these are the simplest membrane transporters. These are located in the outer membrane of bacteria, mitochondria, and chloroplasts. The structure of a porin determines its selectivity. Now, always these are considered always open, so a solute can travel in either direction, and that's just going to be it's going to probably be related to where there's going to be some correlation with concentration of that metabolite. One class or one specific class of porins are known as aquaporins. These are water-specific pores, and these things are truly fascinating. Now, what they do is they allow the transport of water at a very high rate. Now, if you remember, water can diffuse across a membrane, but an aquaporin is a protein that will basically help that water go from one side of the membrane to the other at a very high rate, faster than through simple diffusion. Researcher named Peter Ager was responsible for discovering these. And since then, they've been identified and characterized in numerous different organisms as well as different tissues. Excuse me, here is just a list of some of those. Now, if you look on the right-hand side of this, you'll see the top-down view right here, and then a side view of an aquaporin right here. One of the things that I want you to recognize about this is that there's lots of alpha helices that make up this structure. Now, it's not quite to the extreme of something like hemoglobin, but there's certainly lots of alpha helices. Now, much of the pore is lined with hydrophobic groups that do not interact with water, which facilitates that rapid transport. However, at the tightest point, each water molecule is separated from its associated waters. That point right there to me is truly awesome. Each water molecule, that to me is really fascinating because, it just causes me to reflect on like general chemistry one. When you learn about the concept of a mole, you learn about 6.02 times 10 to the 23rd. And it's almost like, to me, that was always a number I struggled with. I struggled to understand the magnitude of it. Um, but here, what we're doing is this is a protein that basically makes a pore, which is what we see here and what we see right here. And that pore is so small that one water molecule at a time is forced through here. And the cool thing about it is that the amino acids that make up this kind of surrounding region here, 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 and here, they have properties that are going to attract and repel water molecules so that one water molecule, so you can make a single file line of water molecules. Now, the interactions that are going to stimulate this, despite the fact that the pore has lots of hydrophobic residues, there are some arginines and asparagines that are going to interact with those water molecules to get them through one at a time. If this did not occur, rather than water, protons would jump through the channel. So it's these arginine and asparagines that basically help establish hydrogen bonds with those water molecules that get them to go through one at a time. Now, another class of compounds are ionophores. These are an ionophore is an organic molecule that binds an ion, facilitating ion's diffusion through the membrane. Ionophores are often of bacterial origin, and these have been used as as antibiotics because effectively what they can do is they can kind of puncture a membrane and enable ions to move through at a very high rate. They can act as carriers. One example is valinomycin. They can act as channel formers, as is the case with gramicidin A. Both of these, valinomycin and gramicidin A, bind potassium. And what they're going to do is they can only dissipate, or they can dissipate an ion gradient. So ionophores have a very high rate of transport. Now, ion channels. So of the last three slides, your ion channels, porins, and ionophores, I'm most interested in you knowing about ion channels and um, porins, specifically aquaporins. Ion channels are specific for particular ions, sodium, potassium, chloride. Now, potassium channel is 10,000 times more permeable to potassium than to sodium. And that has to do with basically the amino acid composition of the potassium channel and the amino acids that will help bind to, an ion the size of a K plus compared to Na plus. Potassium ion channels can maintain a diffusion limited rate of 10 to the 8th ions per second. So they can move 10 to the 8th ions across the membrane per second. So they're doing it very, very quickly. Now, entrance to the internal pore is lined with 4 anionic side chains. These are going to attract cations, those positively charged cations like potassium and sodium cation. And effectively what these will do, if you look over here in this model, what we have is a protein that, well, here is our hydrated potassium ion. We've got 4 H2O molecules of the oxygens kind of coordinated, pointed towards the potassium ion. And this hydrated potassium will be basically taken in by the protein. Now, this large water-filled vestibule allows hydration of potassium ions. Now, what's going to happen is that this hydrated potassium ion is going to meet where there's this channel, and there's basically a place where a potassium ion can bind right here, and then it gets slowly but surely progressed across to the other side of the membrane. Now, these potassium sites, these are blue or green indicating, or blue ones indicating that it's unoccupied. The green ones are occupied right here and here respectively. So these ion channels, they're pretty fantastic because they can move very selectively certain ions. Now, a next level of ion channels are your gated ion channels. Now, the gated ion channels, what make them particularly unique is that these gated ion channels are normally closed and open transiently to perform specific tasks. The opening and closing is known as gating. So they open the gate and they close the gate. Now, gating occurs in response to a variety of stimuli. Mechanosensitivity, so one of those sensitivities could be, or that one of those stimuli could be contact, physical contact, sound, or other stimuli. They could potentially be ligand gated, so binding of an extracellular ligand, such as a neurotransmitter, might cause them to open up. They can be signal gated, binding of an intracellular signal molecule like CA2 plus, or they could be voltage gated, changes in the membrane potential as the number of cations and anions changed. And in the event that it has, they'll open up. So ligand gated proteins, the way that I think the model that I like to look at are these at the bottom. So nicotinic acetylcholine receptor. These are found at the postsynaptic cleft. And what they do is they bind acetylcholine and they open up, allowing sodium ions to go in. They stay open until that acetylcholine is removed. Now, many neurotoxins and chemical nerve agents affect either the binding of acetylcholine or act as acetylcholinesterase inhibitor or acetylcholinease inhibitors. So this structure right here, it is closed. But whenever this protein binds to whatever ligand, just as an example, whenever it binds to acetylcholine, what's going to happen is that each one of these individual units is going to undergo a rotation. Now, as they undergo rotation, what they're going to do is they're going to form this central cavity between all of them. And that's going to allow sodium ions to basically flood through. We're going to move past this. I'm going to pick up on, we're going to pick up with the GLUT transporters. These are carrier proteins. And I previously described these as they bind glucose, they undergo a conformational change, they release glucose, and then they recover and they're ready to do it again. Here we see that. So here is our open structure of our protein. That open structure of our protein is going to bind glucose. And whenever it binds glucose, then it's going to undergo a conformational change or a transport where the protein will go from this to now like this. So basically that glucose molecule can be dropped off on the other side of the membrane. And then this protein that's no longer bound to glucose will return to its original state so it can pick up glucose and other glucose molecules and move them across. Now, these work both ways. There's not really, when you look at this, it's not to say like, oh, well, this is the cytosol, this is the extracellular space. And if you look at this this model here, it doesn't identify like the inner or outer leaflet. Now, so glucose can be transported into the cell. It can also be moved outside of the cell. GLUT1 through 4 are very specific transporter proteins. They are basically only going to bind glucose. GLUT5 is very selective for fructose. This allows, oh, I apologize. This allows glucose to bind on one side and to be deposited on the other. There's a little bit more to unpack with active transport. Um, Energy input drives that transport. Okay, cool. That good foundation of the phrasing active transport. Some transport must occur such that solutes flow against a thermodynamic potential. Energy input drives transport. Okay, so the energy source and the transport machinery coupled with one another. The energy source may be ATP, light, or concentration gradient. Here, what we've got is a pair of types of transport. We've got this molecule X right here, moving from low concentration to high concentration. So it's going against its gradient. And this particular protein can hydrolyze ATP into ADP and PI. And when it does that, it can harness the energy from that hydrolysis to take X and move them right here. So that's primary active transport. Now, secondary active transport. Well, there's a lot going on with this one. So what we're going to do is we're going to start with X. X is our molecule that's moving from an area of low to an area of high concentration. So X is going to be transported out, and since it's going against its concentration gradient, it needs this hydrolysis of ATP to drive it. Okay, so X gets to the outer side of the membrane and then encounters S, which needs to get to the other side and also is going against its concentration gradient. Well, here the roles have reversed. X moving in is now favorable from the perspective of the, from the perspective of the concentration gradient. S is not favorable, but if X and S can kind of work together, they both end up doing what they want to do. Now, there are some particular proteins that I want you to be familiar with. Those are a sodium potassium ATPase. This is a protein, or this is a protein complex that maintains intracellular sodium low and potassium high. These are crucial for all organs, but especially for the neural tissue, your heart, and the brain. ATP hydrolysis drives NAD out and K in. Okay, so ATP hydrolysis is getting sodium out and potassium in. So it's almost like we've got that secondary active transport, but it's kind of all consolidated. This sodium potassium ATPase is indeed an antiport, meaning that 3 positive charges are exiting, and 2 negative charges are going to, or 2 positive charges are going to go in. So we have molecules kind of passing as strangers in the night. All cells expend a large fraction of their ATP to maintain cytosolic sodium and potassium concentrations. So there's a couple of details that I need you to be familiar with respect to our sodium potassium ATPases. The key to a sodium potassium ATPase is the phosphorylation of a specific aspartic residue on the transport protein. Okay, now if we bounce back for a second, if we then, so this aspartate residue on the transport protein is kind of unique because what that aspartate serves as is kind of the receiver of that phosphate from ATP. So it basically holds on to it in some respects. Um, Now, this ATP phosphorylates the transporter only in the presence of sodium, and the aspartyl protease residue is subject to hydrolysis only in the presence of potassium. Okay, so this is going to be the last slide of the presentation. And this is effectively the last slide of the semester. So this ATPase is this thing right here. That thing is E1. It's also E2. So I don't want you to think about this as like, oh, it's two different proteins. Instead, think about it as it's one protein with two different forms. E1 is the form that is going to bind 3 sodium ions. When E1 binds 3 sodium ions, it's also going to bind ATP. And whenever it binds ATP, it's then going to release the magnesium ion that came along with the ATP. And then, lo and behold, it doesn't release ATP, but rather it's going to release ADP. Where did that third phosphate go? That went on to an aspartate or an aspartic acid on E1. So that residue gets phosphorylated. That is kind of the major indicator whenever E1 is phosphorylated. That's the major indicator that this protein is ready to undergo a conformational change and release sodium ions onto the other side of the membrane. So that phosphorylated E1 undergoes this conformational change. Those 3 sodium ions can then be released onto the other side of the membrane. Okay, so now our protein is still phosphorylated, and what it's able to do is bind to potassium ions. So E2 is phosphorylated and has 2 potassium ions, and at that point in time, as soon as the 2 potassium ions have been bound, now all of a sudden E2, which is phosphorylated, is going to hydrolyze that phosphate away, releasing inorganic phosphate. Then that E2, which is bound to potassium, can undergo a conformational change of its own. And whenever it does that, those 2 potassium ions can be released to the other side of the membrane. So that, again, we can start this all over again, where this ATPase binds the sodium, binds the sodium ions, binds the sodium, then it binds the ATP, phosphorylates the aspartic acid, then undergoes a conformational change, releases the sodium, binds the potassium, releases the inorganic phosphate, moves the potassium to the other side of the membrane, and ta-da. Now, I strongly recommend that you go through this whole figure with a lot of intention and do it deliberately. You can watch this video or this recording and slow it down, scrub back and forth through it. What is this step? Okay, then what happens? Then what happens? It's absolutely important that you know the sequence of this because this is an essential model or this is an essential enzyme for life, but it's also a great model for a couple of different things. I mean, what does this protein right here show us? It's an antiporter. It's going to undergo conformational changes. We are using ATP hydrolysis in order to get things done. So this has a lot of great features and a lot of great traits. At any rate, that's the end of the content for the semester. I hope that this is helpful, and I hope you have a good one.